specifically Professor Siva Sharma, for giving me this opportunity to talk in this wonderful meeting. I just now have submitted my PhD thesis in the Senate of Ben-Gurion University in Israel. And today I'm going to talk about a part of my PhD research, which is about regulation of crack adapter proteins by proline cis-trans isomerases in T-cell signaling. So, two more kind of important key points of my presentation is crack adapter proteins and proline cis-trans isomerases, and I will be briefing on these aspects during the course of my presentation. So that's the simplest way of presenting T-cell activation or stimulation, where a T-cell recognizes an agonist, which could be an antigen, a lymphokine or chemokine, via a cell surface receptor, and initiates a cascade of signaling events, upstream to downstream, inducing certain kind of gene expression, and modulating the cell surface receptor, and producing the chemokine or lymphokine, which decides basically whether the cell will be activated, or it will be energetic T cell, or it will undergo apoptosis. So in these signaling cascades, there are multiple proteins and molecules are involved. Some of them are kinases, phosphatases, and adapter proteins. Now, my talk is about crack adapter protein, and this is a kind of very important protein in development of mammals, and specifically for T-cell activation, it's a critical protein. And we have checked the expression, it's almost kind of expressed, it's a ubiquitous protein. This protein was initially identified, it was an oncogene, viral crack, and then the mammalian crack was crack 1 and crack 2, and crack L, a parallel protein. Crack 2 and crack L are bigger proteins having an extra SH3 domain, which is missing in crack 1 protein. The other part of my presentation is proline cis-trans isomerization. Proline is a residue in nature which can acquire both cis-trans and cis conformation, and both these conformations are energetically favored. And there is a spontaneous transition from cis to trans or trans to cis. However, this takes a lot of energy, around 30 kilocalorie mole. So it's a slow process. So there are a group of enzymes called PPIases, proline cis-trans isomerases, which reduces this energy requirement and accelerates the process of transition from trans to cis or cis to trans. And two important families of proteins, PPIases, are cyclophilins and FKBP. Now, these two proteins are cellular targets of the immunosuppressive drugs cyclosporin A and FK506. And because they bind very specifically to cyclosporin and FK506, the immunosuppressive drugs, these are also called immunophilins. And these drugs inhibit their PPIase or catalytic activity. There's a seminal paper by Putok and colleagues where they identified the substrate motif of immunophilin, and they found that by a far western blot analysis, the proline residue is critical, which is obvious. However, a glycine residue which should be preceding to proline is as critical as proline to be the substrate of PPIase or immunophilins. So when you looked at the signature motif in crack proteins, we have found that crack 2 specifically has two signature motifs in the linker region of two SH3 domains. And remember, this linker region is missing in crack 1 protein. So we suspect that this protein, these two regions are potential substrate of PPIases. So we hypothesize that crack 2 in vivo, in T cell, should be in two populations. One is in trans conformation, where it can interact with all other binding partners, and hence the signal transaction could be enabled. And another in cis conformation, where it's closed, and it cannot bind to its binding partners, and the signal transaction is disabled. And both this conformation regulation is given by PPIases, proline isomerases. So we asked, do PPIases regulate crack adapter proteins in human T cells? Or in other words, whether crack adapter proteins are substrate of PPIases in human T cells? The first experiment we did is to identify whether PPIases associate with crack proteins, and we used a battery of isomerases, and we did a VGS pull down assay, and we found that specifically FKP12, cyclophilin A, and cyclophilin 40 binds to crack 2 protein, but we did not see any association with crack 1. Remember, crack 1 does not have the linker region, which is potentially having the PPIS motif. In different experiments, we have characterized that crack 2 associates with a binding partner C3G at one indicated exchange factor when this association is constitutive. However, more interestingly, we have found that when crack 2 protein is incubated with the PPIS cyclophilin A, the association with C3G increases drastically. And this was confirmed by both GST pull-down assay and the IP with anti-crack. On the contrary, when the PPAs were inhibited by giving the immunosuppressive drug CSA and FK506, we have found the association of C3G decreased drastically with crack 2. However, there was no such decrease was found in crack 1. So we prepared two subclones of Jarkar cells, stable lines, which are overexpressing either crack 1 or crack 2, and we did an IP with anti-MIG. Both these subclones are also expressing MIG, and we found that the association of C3G decreased drastically in crack 2 overexpressing cells when the cells were treated with the drugs. In a different approach, we have used a friend-based study where we used a chimeric crack protein encoded with YFP and CFP in both terminals. So when the protein is open, there should be less FRET. When the protein is closed, the FRET should increase. And we transfected it to the Jarkar cell. Both the confocal and functional analysis, we have found that when the cells are treated with drug, we found a significant increase of FRET value, meaning that in the presence of drugs, when the PPA is inhibited, more and more crack 2 is in close conformation. Migrations and additions are some functional assays which are directly connected with crack association with C3G, and we found that while using Jarkar cells and Jarkar cell overexpressing crack 1 and crack 2 protein, that whenever it is overexpressing crack 2 and crack 1, the migration on both additions are increasing significantly. However, in the presence of drug, the migration significantly decreases in case of crack 2, but not in case of crack 1. Okay. So I'm going to summarize the talk. So we found that PPI is a cyclophilin A, cyclophilin 40 and SPP12 specifically bind to crack 2 but not with crack 1, and we found it in Jarkar cell. And we have found that also PPI increase crack 2 association with C3G, and PPI's mediated increase in crack 2 binding to C3G is dependent on PPI's catalytic activity. When the catalytic activity was inhibited, we found the decrease of association. And also we found that inhibition of PPA's catalytic activity increased the proportion of Cs for close conformation of crack 2 that we have found by FRET analysis. And finally, crack 2 but not crack 1 dependent addition and migrations were inhibited upon PPA inhibition in T cell. So I'm just skipping this slide, and I'd like to acknowledge my supervisor, Professor Noah Kaizako, and my colleague, Dom, and Israeli colleagues for their support. And thank you for your attention.
Maybe for the dinner. Let's move to the second presentation.
Okay.
I now request our chairperson, Professor Ranjan Nath, to kindly present a token of love and gratitude to our speakers.
I think so.
I request Professor S.C. Lakhotia to come on the stage. Now, I request Dr. Vasu Punch to present a token of love and gratitude to Professor Lakhotia. And I request Professor Lakhotia to kindly accept the token of our gratitude. Now, I request Dr. C.R. Satish to come on the stage. Now, I request Dr. Vasu Punch to present a token of love and gratitude to Professor Satish.
No, I don't know him, but just from the paper, I can see that he has not worked that much
Now,
longer
I
to
request
become a
Professor K. N.
professor.
Pandey to come on stage.
Possible, yeah.
Because 2004-5 के पेपर्स ये ज़्यादातर दिखा रहा था उसके बाद के, तो अभी प्रोफेसर कैसे बन गया फिर इतना जल्दी?
Now, I request Professor Vasu Punch to present a token of love and gratitude to Professor K. N. Pandey.
हाँ जी,
I request Professor K. N. Pandey to kindly accept the token of our gratitude.
चलो ठीक है, वो भी खुश हो जाएगा, प्रोफेसर बन गया।
Let's continue with our presentations. Next speaker is